Our next presenter is Dr. Brenda Vincenzi, Director of Medical Affairs from uniQure. And she'll be presenting on a phase 2 multi-center trial to evaluate an intrathecally administered gene therapy, AMT-162.
Great. So yeah, my name is Brenda Vincenzi. I represent uniQure. I'm the medical lead for the ALS ONE study at uniQure. And today, as Fernando mentioned, I'm going to introduce the episode 1 study, which is a phase 1/2 gene therapy study of AMT-162 in participants with ALS ONE. Brief disclosure. Let me just move this. AMT-162 is an investigational agent currently being studied in the treatment of SOD1 ALS. Its safety and efficacy have not been established and has not been approved by any regulatory body. There is no guarantee that investigational agents will receive health authority approval or become commercially available. The agenda today, so I'll introduce uniQure very briefly. And then I'll talk about AMT-162 and the episode 1 gene therapy study, in particular, how does AMT-162 works and how it is administered to participants. And then a brief overview of the episode 1, the study design, and the status update. So uniQure, our mission is to reimagine the future of medicine by delivering innovative cures that transform lives. uniQure is a pioneer in gene therapy. Headquarters is in Amsterdam, with offices in Lexington and in Basel, where I'm located. We have 25 years of success in gene therapy. Back in 2012, we received approval in Europe for Glybera for the treatment of familial lipoprotein lipase deficiency. In 2020, we started the gene therapy study for Huntington's disease, which is currently ongoing. 2022, we received approval for MGH for the treatment of hemophilia B in the US, followed by approval in Europe in 2023. And this year, we entered the clinic with 3 additional programs. So one is ALS ONE. We dosed the first participants last month. Fabry disease, we also dosed the first participant a couple of months ago, and temporal lobe epilepsy that is about to enter the clinic. So AMT-162, mechanism of action and the delivery method. So AMT-162 uses a viral capsid that carries a microRNA designed to suppress the expression of the SOD1 transcript and aim to lower the SOD1 levels and improve, therefore, symptoms of ALS SOD1. Route of administration, as you can see here on the right side. So we start with a simple lumbar puncture, and then we use a catheter, a spinal catheter. We introduce the catheter, which is threaded up to the spinal canal, and we aim to deliver AMT-162 at 3 different levels. Then the catheter is removed out of the lumbar level. Episode 1, so that's the name of the study, is a phase 1/2 multi-center single ascending dose study to evaluate safety and tolerability and exploratory efficacy of intrathecally administered gene therapy AMT-162 in adults with ALS ONE. Here on the right side, you see a QR code. So if you scan that code, you go directly to the clinicaltrials.gov page to get more information about the trial and the centers in the US. We are actively recruiting in the US. We aim to enroll up to 20 participants, both males and females, adults, all genotypes and SOD1. Location, we have identified 11 sites. We are activating sites as we speak in the US. Study duration, we started recruitment, as said, a couple of months ago, and we aim to complete recruitment by 2026. Although the follow-up the safety follow-up will continue for 5 years. Where are the episode study sites located? This is not a comprehensive list, but mirrors what you find on clinicaltrials.gov. You see we have, as said, up to 11 sites in the US. In California, there's the first site that dosed the first participant in October. Active, of course, the other California site, University of California, Irvine, Orange County is active. We also have Mass General Hospital, active, Columbia, New York, and UPenn, Pennsylvania. And we are working to activate the other sites that you see listed here. We keep the clinicaltrials.com page up to date, and as I said, you still find the QR code. So the study design, very, very simple study design. We start with a single ascending dose, 3 cohorts, so 3 doses. We aim to enroll 2 to 4 participants per cohort. And we have an IDMC, so expert that will review the safety data between patients and between cohorts before we are allowed to dose escalate. The idea is that once the single ascending dose is completed, we are going to select the dose that is safe and adequately tolerated. We take that dose and we open a new cohort, this dose expansion cohort. We enroll up to 6 to 8 participants in this cohort with the dose selected from the single ascending dose part of the study. Study design and objective. As all clinical trials, you start with a screening phase. And then if you are eligible, you enroll into the study and you start with this lead-in period. You receive immunosuppressive regimen before receiving AMT-162 as a 1-dose treatment. This is followed by a safety follow-up of 5 years. As said, the primary objective is a phase 1-2 study, so primary objectives are safety and tolerability of AMT-162 in participants with ALS ONE, but we will, of course, look also into exploratory efficacy. Key inclusion and exclusion criteria for the study. These are key, meaning that this is not a comprehensive list. Please also refer to clinicaltrials.gov. Key inclusion criteria. So we ask for a confirmed clinical and genetic diagnosis of SOD1 ALS, slow vital capacity of 50% or above, ALS-FRS-R scores of 25 or above, and of course, participants need to be able to provide informed consent and comply with the study procedures. Key exclusion criteria. We received a number of questions, especially considering tofersen on the market. So exclusion criteria are prior SOD suppression therapy with an antisense oligonucleotide mediators like tofersen. But there is an exception, as you can see here, there is a little star, and refer to the box on the right. There is this exception that participants who previously received tofersen may be enrolled if the last dose of tofersen was received at least 20 weeks prior to the first screening assessment and based on the opinion of the investigator. Any, again, other exclusion criteria, prior SOD1 suppression therapy with viral microRNA mediators or other AAV gene therapy. On the bottom, there is also a further specification. Participants must be willing to forego new ALS treatments through at least 6 months after receiving AMT-162. But after 6 months, if the investigator with the participant, they might decide to add the new ALS medications or change existing medication, which means that even if you are still enrolled, you have received AMT-162, you might start tofersen 6 months after receiving AMT-162, being still enrolled into episode 1. This is my last slide. So, of course, if you want to find more information about the episode 1 study, talk to your doctor, your care team, visit clinicaltrials.gov. You can just go through either the page with this NCT number or just scan the QR code. Thank you to ALS Trial Navigator, AIM ALS. We are listed as episode 1 on their trial navigators. And these are the contacts. If you have any questions, of course, we are always very happy to answer. Thank you. Happy to take some questions. Let me stop sharing.
Thank you, Brenda, for a great presentation. Really interesting technology. I had one specific question about a different therapeutic approach that's out there, and whether people using this treatment could also be in this trial. AP101, a monoclonal antibody targeting misfolded SOD1, is in clinical development. It's in trial. Is that one excluded, or is it only the ASO therapies?
It is excluded. Yes.
Oh, that one is.
Yes.
And then you mentioned the immunosuppressive regimen prior to receiving the treatment. Could you share any more details about what that regimen is?
So it's not public yet, but we
Okay.
we follow a very similar immunosuppressing regimen as in the compassionate use patients that were those. There were two patients, those with a similar AAV therapy from the Bob Brown group. So is we start 2 weeks before, and then we continue for a couple of months after injection.
Thank you. We have a couple of questions in the chat. One is a question of how will you be able to recruit SOD1 ALS patients, given that most will be on Qalsody, or may have tried it in the past and couldn't make it due to tolerability issues?
How are you doing?
So there are actually is a great question, right? We are facing definitely like it's not an easy task like to recruit. We are actually very happy to have tofersen for patients like available on the market. There are a number of participants or patients that are interested in the trial. Either they have tried tofersen before, and the lumbar puncture sometimes, especially if they live very far from hospitals, like going back every month for the LP, might be a little of a burden. So they are looking into something different. Just a reminder, as I said before, maybe for people that are not very familiar with AAV, the idea, one of the main difference, apart from the mechanism of action, is that for episode 1 and T162, there is only one administration. And there are also participants that were on tofersen before. If they develop some adverse events and they were not eligible to continue, the discussion is with the principal investigator at the site. They might be eligible if the principal investigator deems that participant potentially like a good participant for the study.
Thank you. And another question that's specific to your presentation. How far up does the spinal catheter advance into the vertebral canal?
Yes, it's a great question too, but we haven't shared that publicly. Yeah.
Okay. Well, thank you so much for a fantastic presentation. We learned a lot more.